What is somatic gene therapy, and how does it work? Imagine a tiny change inside your body that could help fix a disease at its source. That's what somatic gene therapy aims to do. It's a way to treat illnesses by changing the genes in your body's non-reproductive cells. These are the cells that make up your muscles, liver, blood, and other tissues. Unlike genetic changes that can be passed to children, somatic gene therapy only affects the person getting the treatment. It doesn't alter their reproductive cells or future generations. The process starts by finding the faulty gene causing the disease. Scientists then create a healthy version of that gene or a special gene product that can help. To get this new genetic material into your cells, they use tiny carriers called vectors. These are often modified viruses that can slip into cells easily without causing harm. There are two main ways to deliver the therapy: ex vivo and in vivo. In ex vivo therapy, doctors take some of your cells out of your body first. They then add the healthy gene to these cells in the lab. Once the cells are fixed, they put them back into your body. For example, in some immune system disorders, blood cells are taken out, given the good gene, and then infused back into you. In vivo therapy, the genetic material is injected directly into your body, targeting the affected tissues without removing any cells first. Once inside the cell, the new gene can either become part of your DNA or stay separate. If it joins the DNA, it can produce the necessary proteins to help your body function better. Sometimes, the gene stays outside the DNA, but it still helps by producing the needed proteins. Because your body's cells naturally die and are replaced, the effects of somatic gene therapy might not last forever. You may need additional treatments over time. This therapy is often used for diseases caused by a single faulty gene, like cystic fibrosis, hemophilia, or certain immune deficiencies. Researchers are also testing it for complex diseases like cancer, where genes can help your immune system fight tumors, or for infectious diseases, where genes might boost your resistance. From an ethical point of view, somatic gene therapy is generally seen as acceptable because it treats the individual without changing future generations. It aligns with principles like beneficence, which means doing good, and non-maleficence, which is avoiding harm. Patients must give informed consent, understanding the risks and benefits. Fair access to these treatments is also important, so everyone has a chance to benefit. However, there are challenges. Delivering genes safely and efficiently to the right cells is difficult. There's also a risk of immune reactions or the gene inserting into the wrong place in the DNA, which could cause problems. These risks are carefully considered during clinical trials and treatment planning. In the bigger picture of medical advances and ethics, somatic gene therapy shows how innovation can be balanced with caution. It focuses on helping individual patients while respecting safety and personal choice. As research continues, ongoing conversations among scientists, ethicists, and the public help guide responsible development and use of these therapies. In essence, somatic gene therapy is a targeted way to modify non-reproductive cells to treat disease, always guided by ethical principles that prioritize patient well-being, safety, and fairness.